All right, good morning, everyone. So let me just remind you how this works. This is a student-led review, which means that if you have questions, you are invited to raise your hand or write them into the chat. And then we will just go through whatever you want to talk about. Now, with that said, the small amount of structure that I will impose today, aside from having you sign in, is that I will kind of divide the time evenly between the sections and only move forward if it seems we've run out of things to say. So for now, the structure is that every 30 minutes, we'll sort of switch. We're going to focus initially on the growth topic, and then in 30 minutes or sooner, depending on when we're done talking about it, we will switch to metabolism and so on, just to make sure that we give equal time to all the different parts of the class. With that said, if 30 minutes is more than enough time to talk about growth and we're done at 9:50 or at 9:45, let's say, I'm happy to move on to metabolism at that point. Also, you're invited, if you have questions about the unknown ID process, to stay behind after class. I will also be creating an office hour tomorrow where you can drop in and talk about your unknown ID during the morning hours, probably from like 10 to 11 or something like that, but I haven't quite decided on my timeline. So you'll see an announcement about that go out later today. So with all of that said, if you have questions about, again, what we're going to focus on right now is the growth section, chapter 6. Growth. If you have questions about growth or anything in the growth that you want me to go over, if you want me to review calculations that we covered in class, or parts of the video lecture, or any questions from the study guide about growth, please raise your hand or write your question into the chat and let's talk about it. Kiara, go ahead.
Hi, I'm confused about a question on the study guide. On the study guide, it says which group contains pathogens or food spoilage organisms and why. I understand that it's mesophile and psychotrophs, mesophile containing the human pathogens and psychotrophs containing the food spoilage, but I'm just confused on the why part.
Right. So when I asked this in class, and I sort of phrased it a different way, and I said, well, if you were to predict which of these groups contained the most human pathogens, and everyone guessed mesophile, what was your reason for guessing that it was mesophiles?
I would assume that it's because it's the 37 degrees and that's like where our body's at.
Exactly.
Temperature.
Right. Yeah, that's exactly right. So human pathogens have to be able to thrive at human body temperature. And so it only makes sense that the primary category of, like, if you look at all human pathogens, where do they fall? They fall into the mesophile category because they also thrive at that temperature. So that part makes sense to you, right?
Yes.
Yeah. And as for food spoilage, that typically happens at slightly colder temperatures, room temperature, or even sometimes refrigeration. And so the food spoilage organisms, there is some overlap, but it typically is the ones that cause the visible deterioration of the food can handle, room temperature or sometimes colder temperatures. So does that part make sense?
No.
So go ahead.
So the food spoilage one is it because they can handle lower temperatures?
Yes. And there are certainly mesophiles that spoil food, that's true. But there are also psychotrophs that have no ability to survive in the human body and are not going to harm you, but will make your food look weird and smell bad.
Oh, okay.
Does that make sense?
Yeah, that makes sense. Thank you.
Great question. Okay. Next question is from Paige. Go ahead, Paige.
I don't know if it's something we're going to need to know for the test, but are we able to go over the nitrate test, like the nitrate reductase and the nitrite reductase?
Yes, let's do that. What you would more for the test, you wouldn't have to remember like the detailed procedures, because I usually don't require that And if I do, I will make a big deal out of it, like I did with the gram stain and the endospore stain and
Yeah.
stuff. So you don't have to know the procedures, but it is useful to understand what that test is telling you. And
Yeah.
that is to say that nitrate is being reduced, right? So what does it mean to be reduced?
It's gaining an electron.
Exactly. And what context, what might that be happening? When an organism is putting electrons onto nitrate, what sort of metabolism is likely to be happening that would make that useful to do?
Anabolism.
Say again?
Anabolism.
It's not, it is a form of catabolism. It's one of
Sorry,
the major ones, like do you remember anaerobic respiration when we talked about that?
Mm-hmm.
And how you have to put electrons onto something that's not oxygen?
Yeah.
Yeah. So it's not the only time that nitrate is reduced. So microbes sometimes reduce nitrate for other reasons, but it's a common one where in an organism that does anaerobic respiration, if its way of doing that is by putting electrons onto nitrate, then it would have nitrate reductase and be able to reduce nitrate. And you'd see that in the nitrate test.
And then,
Go ahead.
but then, so what about the nitrite?
Yes. So the first, yeah, and I'm happy to go over the whole test. I'm just, yeah, and I totally,
it's mostly just the concept that confuses me. I don't understand what's exactly what it's telling us.
So I'll go through that that whole test again, but just to sort of put it in context for what you need to know for the test.
Mm-hmm.
You don't really have to know, like, you don't have to remember that nitrite is the reduced product of nitrate. You do have to understand why nitrate might get reduced. In what context. Like if I was to say that something was positive for the nitrate test, then I might reasonably expect you to be able to at least suspect that it might be capable of anaerobic respiration.
Okay.
But with regard to what you're talking about, I'll briefly mention it because it's more the details that you're, I think, getting at have more relevance to the Unknown ID project.
Yeah, that'd be
so very briefly, what I'll say is that the first thing that happens when you add electrons to nitrate is it turns into nitrite. And that's, so if nitrite is present, like if you give an organism nitrate, like we did in the nitrate test, we put it into a broth that had a lot of nitrate. It had no nitrite, it had nitrate, which is NO3. And if you put an organism into a broth that contains nitrate and it turns it into nitrite, it had to do that by adding electrons to the nitrate. So we can tell that it's reducing nitrate. Some organisms continue to add electrons to nitrite, and that turns it into either ammonia or nitrogen gas, which is a more reduced form. And that's why the nitrate test has two components. You first have to test to see if the first step happened, if it went to nitrite. And if it did, and you can see that, you're like, oh yes, it did reduce nitrate. Does that make sense?
Yes, but then does that mean it just hasn't been, it doesn't reduce nitrate?
If what?
If it just goes to the nitrite?
If it goes to the nitrite, it means it did reduce nitrate. So when you put electrons onto nitrate, you turn it into nitrite.
Okay.
Nitrite is the reduced product of nitrate reduction.
Yes.
Does that make sense?
Kind of, yes.
Kind of.
That makes sense. I think I'm just having trouble with the, so some organisms can reduce it to the nitrate and then others can also reduce it to the nitrite.
So nitrate is the starting material.
Yeah.
And if an organism puts electrons onto nitrate, it turns into nitrite. Like it definitely does. It always does.
Always does. Okay.
Yes.
Always does.
And so if you can tell that that's what happened, like when you do your first step in that nitrate test and you
then
find nitrite, it's definitely reduced it.
That makes sense.
However, if you don't find nitrite, you don't know what that means because some organisms then keep adding electrons to nitrite and turn it into something else. So the fact that there's no nitrite either means that no nitrite was made at all, no reduction happened, or it means it was made but then turned into something else. And that's why you have to do the second step.
That's okay. That's what I was confused about.
Does that make sense though?
Yep.
Awesome. Okay, great question. I'm going to check the chat because it looks like there's a couple of relevant questions. And let me first go to Lauren says, is nitrate reduction mean it's doing fermentation? No, that's a great question, Lauren. And fermentation is a very separate process. So what happens in fermentation is that the electrons that are pulled out of glucose during glycolysis are put back onto pyruvate or a derivative of pyruvate. But either way, it's an organic molecule, a high energy organic molecule. And really the electrons that are put back have not been bled of any energy. They're basically the same electrons that you started with. You had to pull them out because that's just how glycolysis works, but you don't use the energy of those electrons, you just put them back, you waste it. And the product of fermentation is always an organic molecule, usually an organic acid, but not always, right? Could be ethanol, which is not an acid. So nitrate reduction is respiration, which is a separate process. And respiration always means you went through glycolysis, but then what you did with pyruvate was then you put it through the Krebs cycle. And then the electrons gathered during glycolysis in the Krebs cycle all went to the electron transport chain. That's respiration. Nitrate reduction can be a form of anaerobic respiration because when you go through the electron transport chain, the electrons have to go somewhere, right? And they end up in aerobic respiration on oxygen, but in anaerobic respiration, they have to be put somewhere else. And nitrate is an alternative to oxygen. It's an inorganic, low energy molecule, right? Not an organic, high energy molecule. Great question. Okay. And it looks like Kiana, I think Kiana, if you're still there, go ahead and unmute, but it looks like you might have disappeared from my chat. So I'll just wait 1 second to see if you're there, Kiana. Okay. Alex has a question. Go ahead.
Hey, Hillary.
Hey.
I have a question. I just wanted to touch briefly on transformation efficiency. There's a practice exam on Canvas that I did, and on number 4, I'm confused how we got the first 2 answers. I don't know if you can pull it up, but I understand how to get I understand how to do all of it when you're given the DNA concentration and the volume, and then that gives you the micrograms of DNA.
I'm trying to pull it up right now. Hang tight. And well, I won't ask you this question until I have it up, because if you answer, I'll have to have you just repeat the question because I won't remember. Okay. All right. I'm going to do a preview so that we can't see the answers. Okay. And so you're talking about this question here.
Yeah, the total amount of DNA you added to the 2. It
Yeah.
says it's. Do you want me to say the answer?
Yeah, go ahead. This one's not for credit anyway.
Okay, it's 1 microgram, and the second one I think was 1 milliliter.
So yes, that so far is correct.
So how did we get those 2 answers?
Oh, for part 1 and part 2, you found by hunt and guess what they were, but you don't know why they are the way they are.
Yeah.
Got it. Okay, perfect. Thank you. All right. So yes. So how do you get the total amount of DNA? So in this tube, what you added was a volume of DNA, 2 microliters, but this is a volume. It's not an amount, right? To find the amount, you have to know what the concentration was. Right. So this was the concentration right here of the stock solution.
Okay.
And then we took 2 microliters of this stock solution. So an amount is always concentration times volume.
Right.
Does that make sense?
Yes.
Yeah. So if each microliter of this stock has 0.5 micrograms, then 2 microliters would contain 1.
Oh, got it. Okay.
Right?
Yeah.
Okay. And now the total volume is, you started out with a tube and in it you put 500 microliters of this transformation solution. Then you added your DNA, and then next you added broth, 500 microliters of broth. So 500 plus 500 is 1 mil.
Okay.
Make sense?
Yes. Thank
Sweet.
you for clarifying.
Any other things about this, or do you want to save any other DNA questions till a little later?
I think the rest of it I've got, so we can save the rest for later.
Okay, sweet. All right, so let's see who is next. Paige, did you have a second question?
I had a question about, or can we still do the transformation efficiency now, or should we, or we wait till later?
I don't see other questions in the chat, so or hands up. So as long as this is where we're headed, and guys, just thank you for asking that, Paige. Just so you know, the rough kind of outline of this is that we're spending the first 30 minutes on growth, and then after that metabolism and so on. So if you have questions about growth, go ahead and raise your hand. But let me address Paige's transformation efficiency question first, and then we'll move forward through growth as well. And I see, Kiana, that you're back. Great, and your hand did go up, so I will get to you. Thank you. Paige, what is your question? And did you want me to just go back to this quiz?
Not the quiz. I have a question because so for when it says total, if I'm remembering correctly, with the lab, it was like, sorry, I'm trying to figure how to word this. When it says total volume, there was a question where it was like 250 and then 250, and then the DNA was like 10.
Mm-hmm.
But I don't know if it was on the, I think when we were talking about it in class, or maybe it was on the review, it didn't include the DNA in that total volume.
Right. Yeah. And so I mentioned this in lab as well. And so, yes, if you had this reaction that you just described, and this is actually the one that you did in lab, technically the total volume is 510. I said, if you were to round to 500, it's not going to substantially change anything, and it's easier math. So go ahead. And on a test, I would encourage that too, because you're not trying to get like, specific digits or anything. You're just trying to get the answer that's the closest to what's offered. And so it's not going to change things enough to really worry about.
Okay, cool. That's what I was confused about.
But that makes sense?
Yes, that makes sense. Thank you.
All right. Looks like Chris is up next. What's your question, Chris?
Hey, I have a few probably quick questions. The first one is in relation to, on the study guide, there's a question that says, what are the different forms of media and what are they used for? The deep media, is it used just for like anaerobic bacteria? Because I have down that it's for specific types of experiments, but I don't really know other than anaerobic bacteria what it would be used for.
Yeah, and we didn't cover this a ton. So one example of a deep was the thioglycolate deep, and that one you could use for oxygen preferences. Actually, not the only one. There's a number of them. For instance, motility tests are technically speaking a deep. And that's in your micro test reference. And some of you will have done the motility test and seen the results. And maybe you didn't think to yourself, oh, that's a deep. And all it means, like the technical meaning of the word is just that you poured an agar-based recipe. So something that is going to harden eventually. You poured it into a tube, but you didn't tilt the tube to make a slant. Instead, you just left it like up and down so that the top is flat. And it's deep. it's got depth.
Right. Okay.
So you can use that for lots of things. We used it specifically for the thioglycolate. Some people used it for the motility, but there's other reasons you could use it for. So you wouldn't be asked questions that you couldn't answer based on what we've talked about. What I would expect you to know, probably, if I was to ask a question about media, would be something like, would you use a Petri dish or a broth if you were trying, or like, would you use a broth or a slant if you wanted to get rapid growth that you didn't have to keep? And so the broth is what you use when you need a ton of organisms really fast and you don't care about keeping them later. And the slant is what you use if you're going to have longer term storage. So that's the emphasis really. And the Petri dish, of course, is the one you'd use if you're trying to get like single colonies or something like that, or to see to see a result like a selective or a differential media. It's a lot easier to see on a Petri dish. So sort of straightforward questions like that, nothing where you'd have to rack your brain and go, well, she didn't really talk about deep, so I don't know what to say here.
Okay, cool. Awesome. The other question I have is a few questions down there where it talks about the PEA, MSA, and blood auger.
Yes.
So the blood auger, we kind of briefly went over it in the lecture that you had, but just to make sure that I fully understand it, there was a diagram that was used that had gamma, alpha, and beta hemolysis. The gamma hemolysis means nothing happened, correct?
Yes.
And then the beta hemolysis, which is kind of on the other end, means that the bacteria was able to like explode the red blood cells and use the nutrients from them.
Yeah.
And I'm assuming that alpha just means that it partially broke them down and it didn't fully break them down.
Yeah. And
what does alpha mean?
It's a different kind. It's like the red blood cells don't explode completely or as much. And so you get sort of more of a hazy background, and we really
Right.
don't talk about that. So the distinction will be much more clear cut. Either they exploded the blood cells or they didn't.
Okay, cool.
So
All right,
it's
thanks.
a mostly useful test to determine if something is capable of beta hemolysis, which is what makes it more dangerous as a pathogen.
Right. Okay. Awesome. Thank you. That's all I
You're
had.
so welcome. All right, let's go next to Kiana. You've been so patient. I'm glad to hear that your Zoom is cooperating with you. Go ahead.
Yeah.
What's your question?
Yeah, sorry, my computer just froze and I was like, what is happening? But
It's so frustrating. I've been there. Oh, it's the worst.
yeah, I have a question on like the categories of organisms. It's kind of towards the top of the study guide, but it's like, which one is based on carbon and energy sources?
Oh, great. Yes. So that one is do you remember when we talked about heterotrophs and autotrophs and photoautotrophs and chemolithoautotrophs, all that stuff? So,
Yeah.
so just to briefly summarize, autotroph, what that means, that's the hardest one for people to remember. Autotroph means that they're capable of making sugar from scratch, basically. So their carbon source is carbon dioxide. They take CO2 out of the air and use it to make organic chemicals. So autotroph always means that they're taking CO2 out of the air and making organic chemicals. Carbon fixation is another term for that, but that's what autotroph means. It technically is derived from it means auto or self-feeding. So they're able to make their own sugar, basically. Heterotrophs have to take the organic chemicals out of the environment that some other organism originally made. Does that make sense?
Yeah, that makes sense.
And
Okay.
then in terms of the sort of other part, like what's the energy source? It could be in the case of heterotrophs, the energy source is the same as the carbon source, so they usually just refer to them as heterotrophs, although technically we would call them chemoheterotrophs, because chemo is the part where it says, where did the energy come from? Chemo means the energy came from chemicals. But since the carbon source is organic chemicals and the energy source is organic chemicals, we just call them heterotrophs for short. However, when we talk about autotrophs, there's photoautotrophs. They get their energy from
The sun.
right, but their carbon is from CO2.
Mhm.
And then chemolithoautotrophs get their energy from basically metals or rocks, and their carbon from CO2 because they're still autotrophs.
Okay.
Does that make sense?
So most of the things just get their carbon from the CO2. It's just how their energy comes from that's different.
The autotrophs get their energy, the autotrophs get their carbon from CO2. The heterotrophs get their carbon from organic chemicals in the environment.
Okay.
And then those autotrophs are subdivided into, are they photoautotrophs getting the energy from the sun, or are they chemolithoautotrophs getting their energy from chemicals?
Okay, perfect. Thank you.
Okay, great question. Let's see. I have some in the chat as well. So let me go and look at that. Inessa asked, what's the chemical formula for nitrate and nitrate? Great question. So let me just screen share my little thingy here. So nitrate is NO sub 3, NO3, and nitrite is NO subscript 2. Does that help?
Yes, thank you.
Awesome. Okay, Kiara said, I have a question for microbial growth. How is the gaseous environment controlled to grow anaerobes and microaerophiles? And so that's out of the study guide 2. Let me screen share that. I think. Okay. I think it's maybe a paraphrasing of a question that's on there. But basically, so it's slightly different depending on how strict of an anaerobe it is. But there's several ways to grow anaerobes. because remember, anaerobes, obligate anaerobes require no oxygen. Facultative anaerobes are fine either way, but obligate anaerobes require no oxygen. And so you can grow them a number of ways. You know, a simple way to grow them, it would be you could grow them in a thioglycolate deep, they'd grow at the bottom. If you want to grow a large amount of them and you want to do it with expensive equipment, you can get an anaerobic chamber. So it's kind of like a big plastic box, like maybe the size of your desk. And they pump all of the air, they pump all of the oxygen out of it, basically. They regulate it with, it has its own air supply, and they regulate the gaseous environment by taking stuff out and putting other stuff in. That's another way to do it. Another way you can do it is with a candle jar. That is done more often for capnophiles that also like a little bit of CO2. And so for a candle jar, you just take like a big jar, like imagine like a spaghetti sauce jar that's empty, and you put your plate inside of it, and then you stick a little votive candle on top, and you light the candle and you close the jar. So what happens is that the candle uses up the oxygen, right? Because it's burning, and that it needs oxygen to burn. And as it's burning, it releases CO2 into the environment. And it's a sealed container. And so we have these in lab. They're kind of like a really ghetto version of an anaerobic chamber that's really expensive, and the candle jar is really not expensive. And another way to do it is in a bag. So you take like a plastic bag, like a Ziploc, but it's much tougher. You stick the plate inside the Ziploc bag, and you stick in a little pouch that has a reducing agent that basically puts electrons onto oxygen. So it uses up all the free oxygen in that tiny little bag, so there isn't any left to be used in respiration. And then you effectively have an anaerobic environment. In terms of for the curriculum that you might need to know on a test, all you would need to know is that it is some kind of equipment that deprives or changes the gaseous environment to remove oxygen, or to effectively remove free oxygen. Does that help, Kiara? Great. Okay, I'm going to stop share this. So we have 3 minutes officially before we click the dial and go into metabolism. If anyone has a last minute question about metabolism, now is the time. It's not that you're not allowed to ask metabolism questions if some occur to you later, but we are trying to impose a little bit of structure on this. So if you have a burning growth question right now, go ahead. I think I maybe misspoke that. If you have a burning growth question right now, please raise your hand or put it into the chat. I'll give you a second to think about what that might be. Okay. Christina asked if we can go over the growth calculation. Absolutely. Okay, so let's see. One way we could do it is, I think I put an example in here into the, we'll just start with the example in the study guide. Okay, so this is a straightforward one. This is actually one of the more difficult ones. So let's do this one. Why not? So this is the one that I think most people have the most difficult time with, which is finding the generation time. For some reason, it's easier to do the how many cells did you end or start with, but finding the generation time can be kind of tricky. So we'll do one of each. Let's start with this what I would consider more difficult one though. Okay, so we have this guy, and maybe what I'm going to do is share my whole screen, because I've got a couple of different windows open where I'm going to write things, and that might help us better follow along. So don't judge me. I have lots of windows open. Okay, so here we have this guy, and what happened is, let's see if I can make my text a little bit bigger. He ate this burger that at the beginning had 25 cells in it. So this is like, remember the starting number is 25 E. coli cells. But by the time he ate it, there were more. So it sat in the passenger seat in his car after he ordered it for a while. And then he went and ate the burger. And at the end, when he actually ate it, there were 12,800. So what's the generation time? So how do we go from 25 to 12,800 with the little and sub-zero? Sorry, I don't know if you guys can hear the family situation in the background. We've got children running around with dinosaur hunts and things like that. It's pandemonium here. All right, so this is X. We want to figure out first how many cell divisions happened, because here's the thing: to know the generation time, we need to know how many cell division happened. Right? We know the incubation period, that was 3 hours. So we know there were 3 hours in which 25 cells turned into 12,800. So this information is going to allow us to figure out how many cell divisions happened. And once we know that, we're going to be able to divide this time period to find out how long each one of those cell divisions occurred, how long it took. Hopefully that made sense. Okay, so the first thing we can do is we would take our ending number divided by our starting number. And because that's going to be 2 to the power of X. And then that number is, I don't know where I put my, there's my calculator. Hopefully you can see my calculator. $512, okay? 512 is 2 to the power of X. And then we just figure out what X is. And so if you have the right calculator, you can do this. You can actually, the calculator will figure it out for you. But for many of us, that might not be the case. And so you can just, put different numbers into there until you can either just do it the hard way, which is to say, it's the easy way, but at the hard way, you can go 25 goes to 50, goes to 100, goes to 200, goes to 400, goes to 800, goes to 3,200, goes to 6,400, goes to 12,800. Right? That's one way to do it. And that didn't take me very long. But you could also just substitute numbers into here until you figure out that the answer is, I'm just going to make these little things a little easier to see. 1, 2, 3, 4, 5, 6, 7, 8, 9. This was 9 cell divisions. You can also use a calculator that has this function on it where it will figure out what X was for you, or you can use a calculator that has a exponent function and just put numbers into here until you get to 512. Before I go on, let me just pause for a second. And I will say one thing and then I will ask a question. First of all, I'm not going to give you 9 cell divisions on a test. Okay, I'm not going to do that to you. It's going to be less. However, did you understand in principle, yes, no, or kinda, how I got 9 from this?
Yes.
Okay. Let's have everyone else kind of weigh in who's listening in the chat, just yes, no, or kinda. Okay, good. Okay, so now that we know it's 9 cell divisions and we could use a calculator, if we know how to do that, go ahead and do that. That's, that's faster than this, although not a ton faster, honestly, and it's not necessary to have that special feature on your calculator. But once we know that it's 9, we take our 3 hours, which is also 180 minutes, and we divide by 9, and the answer is 20 minutes. So did that make sense?
Yes,
Okay.
it does.
Awesome. Okay, so that's one example. Now let's do a different one. We're going to actually stick with this guy, but we're going to ask a different question. We're going to change some of the numbers. So we're going to say, what if we didn't know the ending number? Okay, but we did know the generation time. And let's say it's sitting in it, he takes the same burger, like let's an alternate universe. Okay, so we have a starting number of 25, alternate universe, starting number of 25, but in this universe, we don't know the ending number. Also, he sticks it in his trunk and it's a little cooler there. So actually maybe the generation time, which is given to us in this example, is a little bit slower. Maybe it's 30 minutes instead. Okay. So let's suppose that we start with 25. We give you the generation time, which is 30 minutes. And we want to know the ending number after 3 hours. Okay. So in this case, what we would do is, again, the simplest way to do it, you can use the equation, and that's fine. And I'll show you how to do that. But the simplest thing is to say, well, 3 hours, which again is 180 minutes, divided by 30 minutes gives me 6 cell divisions. And so 25 goes to 50 in the first cell division, goes to 100 in the next cell division, goes to 200, goes to 400, goes to 800, goes to 1600. And so at the end, it's 1600. And so the answer would be, the ending number is 1600. And we could also answer this fun question about the infectious dose, right? We could say, well, if the infectious dose is 190,000, then this is well below that. So did everyone kind of understand how I did this calculation? Yes, no, or kinda? Go ahead.
I just have a quick question on the counting part. When you said 20 to 50 to 100, are you counting by like every, because I'm not sure how you went from 25 to 50. Are you adding 50 each time?
No, I'm multiplying by 2 each time.
Oh, okay.
Every one of these 25 cells in that 30 minutes multiplies, divides in half.
Oh,
and that gets you to 50. And then every one of those 50 cells in the next 30 minutes divides in half. That's what gets you to 100, but it's multiplying by 2.
Mm-hmm. Okay.
So if you want to use the equation, it's the same thing, but the equation might be more simple for some people to remember. What we want to know is what's n sub t. And we know that n sub t is going to be n sub 0, which is 25 times 2 to the power of x. We figured out what x was is 6, right? There's 6 cell divisions. X is always the number of cell divisions. So 2 to the power of 6. But I'm doing that here, right? What I'm doing is I'm doubling,
Right.
doubling. So that's 6 doublings. That's why this is what it is.
Okay.
And then you would go to your calculator and go 2 to the power of 6 if it has exponents. And if it doesn't, I would just go 2 times 2 times 2 times 2 times 2 times 2 times 25, which is basically what I did here.
Right.
Does that make sense?
More simplified. Yeah. Thank you.
Yeah. Great question. Okay. So go ahead. Sounds like someone had a question, or let me just circle back to Christina. Christina, does that kind of cover what you wanted to look at?
Yes, it does.
Perfect. And I'm sure many people are happy that you asked that question. Inessa, you had a question, your hand went up. I don't know if I've already answered it.
Just to confirm, so the answer for the first part is 20 minutes, right?
Yes.
My quick question, sorry, I don't know if I'm talking over somebody.
Oh, no, go ahead, Sharon.
Okay, my quick question is, I just got a little confused where we had to get the 512. I'm wondering, why do we get the 512?
Oh, great. Okay, so let's go back to this right here. So I was working off of this equation. Sharon, does this equation look familiar?
Yes, it does.
Great. And you know, the different components are like, this is the ending number of cells, this is the starting number of cells, and X is the number of cell divisions.
Yes.
Great. So this is one step after basically I put all the numbers in. So the ending number is 12,800. The starting number is 25, and that would be multiplied by 2 to the power of X. And we want to solve for X. And when we want to solve for something, why I keep putting a 2 in there, then what you can do is you can divide both sides by 25.
Yes.
Right? So you could divide this by 25, and you could divide this, oh, Lord have mercy. The auto functions on my computer are driving me bananas. So you can divide this by 25, and then you also divide 25 by 25. You know, you divide each side by 25 because you know how to do that, right?
To isolate the 2x.
Right, exactly. And so that ends up being 2,800 over 25. And that number is 512.
So what is the number? That is my question.
What is what
does
number?
the 512?
512 is what happens when you take 12,800 divided by 25.
I think what she means is, sorry, I don't mean to interrupt, but I think what she means is, why do we need that number?
Oh,
Yeah.
yes, because we're trying to solve for X, right?
Mm-hmm.
So 512 is equal to 2 to the power of X. So you just now have to figure out what X is. So if you put in, if you put a 2 in here, 2 to the power of 2 is 4. So that's not big enough. 2 to the power of 3 is 8. So that's still not big enough. 2 to the power of 4 is 16. 2 to the power of 5 is 32, and so on. And it's not until you put 9 in here that you get 512. And that's how you know it's 9 cell divisions.
Oh, okay. Now I understand it. It's very clear. So what we are trying to make sure is the number of times the cell is dividing must add up to this total number.
Right. So this is basically, if this is the number that we would multiply by if we'd started with 1 cell, but we started with 12, we started with 25.
Oh, okay.
Yeah.
Okay. Thank you.
Okay, let's see. And then, Inessa, did I completely answer your question?
Yes, I
Okay, good. Okay, just making sure. All right. Any other questions about growth before we officially pass the baton to metabolism? I mean, we've been answering metabolism questions all along, and that's fine, but I just want to check in and make sure we haven't missed anything that anyone wants to particularly review about growth. Okay, if anything happens to you, if anything occurs to you, feel free to raise your hand and ask about growth. But let me just sort of give you the official prompt to, if you've been hanging in there waiting to ask patiently to ask about a metabolism, now's the time. So if you have a metabolism question like, what's the difference between this and that, or how come this is the way it is, or what's this answer to this on the study guide? Paige, go ahead.
On the study guide, it says something about trace the carbon and the electrons, like what's the flow of them?
Yes.
And I just want to make sure that I understand that correctly. Like so are we doing, sorry, let me find it in my thing real quick, because I just want to
I got it. Here we go. Yeah. So you're asking about this, right? What's the flow of electrons during these processes? What's the flow of carbon?
Yeah. And I just want to make sure, well, I want you to explain it, but I want to make sure, like for respiration, do you mean like it goes from the food and then the NADH and the electron transfer chain and the oxygen?
Yes.
Okay.
I'm going to screen share. And so in case that was cryptic to other people, I will just elaborate. So let's start with the electron flow. It's not sort of an official, there's not just one answer to this, but what I'm trying to get you to think about is, in previous courses, you were taught about metabolism, like glycolysis happens, the Krebs cycle happens, the electron transport chain happens. And at some point in the future, you may even have to know all the specific metabolic intermediates, their chemical names, and all the specific enzymatic names, depending on how far you get into whatever field you're going into. But sometimes knowing just sort of like the big picture about these are the 3 pathways, or the really minute details, obscures what's really important. What's really important is why we're doing this. The way to get energy out of glucose is to break it and steal the electrons out.
Yeah.
Right? And then put it into the electron transport chain. That's the whole point. So all the details are whatever, but we can say we can sort of surface this or emphasize this or learn this by saying, okay, where are the electrons? It's kind of like a shell game, like where are the electrons? At the beginning, the electrons are on the molecule glucose. During glycolysis, the electrons end up in 2 places. Some of them are removed from glucose and they go to NADH. Some of them stay in the original organic molecule in the form of pyruvate, because pyruvate still has some of the original electrons from glucose. It hasn't been fully scavenged yet. And then from there, the electrons go the pyruvate electrons go to the Krebs cycle and end up on NADH, so more NADH, right? And then they end up going through the electron transport chain, and then they end up on oxygen, which turns into water. So there's not just one way to diagram this, but if this is respiration, right? Let's compare what happens to electrons during fermentation. In fermentation, the electrons are still starting out in glucose.
Yeah.
And they do go to NADH, and then they go back to pyruvate, which turns into whatever it turns into, sometimes lactic acid. In anaerobic respiration, where the electrons, they start out in glucose. They go to NADH and pyruvate, more NADH, electron transport chain, and then nitrate or carbonate or sulfate or whatever, and then it turns into whatever the reduced product of that is. Does that make sense?
Yes, that makes
Okay.
more sense. I just wasn't like, I don't know, it was either worded weird or my brain doesn't work.
It's not you, it's me. It's not you, it's me. I'm asking you a hard question that you cannot find it in a textbook, because
Okay,
what it is basically is asking you to think about like this from a different perspective than just how you're taught or what you memorize. Same with carbon, the carbon flow. So where does the carbon start? Let's do a respiration first. The carbon starts in glucose. Then it ends up in pyruvate. Then it's all throughout the Krebs cycle, but also turns into CO2, right? Basically the end of it, all of it's CO2. All the carbon goes to CO2. And we've just taken like a big enzymatic jackhammer and just pulverized the crap out of the glucose until it's splintered into all the CO2 pieces. That's where all the carbon is. Does that make sense?
Mm-hmm.
Yeah. And we could ask different questions about that I'm not going to, hopefully, if I was to ask you a question like this, it would be relatively straightforward in terms of if you understand where the pathways are, then you can figure it out.
Okay.
Okay.
Yeah.
Great question. All right, let me stop share and just check my chat first, and then I see 3 hands up, so I will go in the order received. But I just want to make sure I'm not missing out on anyone who needs to weigh in on it in the chat. Okay, great. Kiara is next. What's your question, Kiara?
Hi, there was this question on the study guide. It was like, what are the chemical and biochemical features?
Okay. Kiara, let me just make sure that your audio cut out, so feel free to weigh in the chat. Oh, can I hear you now?
Can you hear me now?
I can hear you now.
Okay.
I found the question you're talking about. Is this it? What are the chemical and biochemical features of enzymes? Yes. Okay, so first of all, enzymes are proteins, so you need to remember that. They're therefore sensitive to pH and temperature. And then they can be inhibited by chemical inhibitors, non-competitively or competitively. The competitive ones inhibit them by binding competitively to the binding site, active site, and the non-competitive ones bind some allosteric site.
Yes.
And you have to remember that enzymes have an active site and that they facilitate chemistry, but they themselves are not destroyed by the reaction or affected by it particularly.
Yes.
So all those basic things.
Yes.
Great question. Anything else? Does that completely answer your question about that?
Yeah, that's it. Thank you.
Okay, great question. Sherry, what's your question?
Okay, so my question is from one of the review quizzes we did. This was for the metabolism section. I'll just post a little screenshot of that question, why I'm confused. I want to know why the last option I picked was incorrect.
Okay, so.
Because it was pretty broad.
All right, so which of the following are unique features that both chloroplasts and mitochondria share and which distinguish them from other organelles? Uh-oh, what happened to my screen share? Something weird happened, hold on. Did I do that? Okay, there we go, we're back. All right. Okay, great. So you picked a lot of good answers, right? They both have two membranes. They both have their own DNA and ribosomes. They both use electron transport chains to harvest energy from electrons. They both generate ATP, and all those are accurate. Only one of them generates ATP by harvesting energy by breaking chemical bonds in organic chemicals. If only one of them does that, what would be your guess as to which one?
Um,
And if you have no idea, say that. That's fine.
yeah,
No.
I would guess both still. I'm
Okay.
not sure.
So let's remind ourselves what's happening. Now we have to sort of think about what's the metabolic process that's happening in each of these organelles. So in mitochondria, what you're doing is, pyruvate's coming in through the Krebs cycle, right? So pyruvate is an organic molecule. It's got, 3 carbons and hydrogens and oxygens and whatnot. It's a high energy organic molecule. You're ripping off one of the carbons, throwing it away, getting some electrons out, putting into the Krebs cycle, ripping off carbons, throwing it away as CO2, getting electrons out. So you're harvesting energy by breaking chemical bonds in an organic molecule that was pyruvate, and before that was glucose. So that part makes sense, right?
Yeah.
Okay. What's happening in the chloroplast is not that, right? The energy in the electrons is not coming from electrons harvested from high energy organic chemicals. The reason that the electrons are energized is because we stuck the electrons into a photosystem and we heated it with the sun. So the energy is coming from the sun. Does that distinction make sense?
So the one that would be harvesting energy from breaking chemical bonds would be the mitochondria then.
Exactly. And it's not both. It's not both.
Got it. Okay. Thank
Does
you.
that make sense?
Yeah. The sun part made it clear.
Excellent. Excellent. And thank you for asking that. You are very likely not the only person who is like, gosh, why? Why isn't this the right answer? Okay. So Kiara Shepherd, because I have 2 Kiaras and I have to remember to distinguish them, asks, could we go over this question as well? Please understand that fermentation reactions differ between organisms based on genetics. Yes, absolutely. Kiara, let me go to the study guide so other people can read the question, kind of follow along. Good. Okay, so let me get my screen share up here. Boom, boom. Okay, so the question or the prompt in the study guide is this. Understand that fermentation reactions differ between organisms based on their genetics. Okay, so fermentation is not just one thing, which is maybe a little bit different than like aerobic respiration. Aerobic respiration is one thing. Like if I say aerobic respiration, it's like all of glycolysis, all through the Krebs cycle, the electron transport chain, we go to oxygen. Like that's all fairly set. There's a few variations of that, but it has to do with the specific name of the specific enzyme. But fermentation is a broad category of reactions. They share a lot of things in common. They all start with glycolysis. And that means that glucose is broken into 2 pieces that end up being pyruvate. And in the process, we take some electrons out and put it onto NAD+, generating NADH. So that part's the same. But after that, the next reaction or reactions differ based on what the organism's enzymes are. That's genetic, right? Genetics determines what enzymes you have. So if it's an organism that can take pyruvate, dump electrons onto it, and produce lactic acid, then it will do lactic acid fermentation and produce lactic acid as a byproduct of fermentation. If it's an organism that decarboxylates pyruvate and then dumps the electrons onto that decarboxylated form of pyruvate, making ethanol, then it does ethanol fermentation, generates acid and ethanol, or not acid, gas and ethanol. If it's an organism that has a different enzyme that takes pyruvate and reduces it, but does some other funky stuff to it and generates a different organic acid, we will get a different thing like acetic acid instead. Kiara, does that make sense? Yeah, okay. All right, so also we have a question from Lauren. Lauren, go ahead.
Mine's actually very related to what you just spoke about. My question was about know the details of the lactic acid and ethanol fermentation reactions.
Great.
And what I'm looking at, I have my book open, and what you just said is that in, oh wait, so in one of them, the pyruvate is decarboxylated and reduced into ethanol, but then the pyruvate in the lactic acid is not decarboxylated, or whatever that term is. It's putting the electrons from, or it's adding electrons to NADH. I'm very confused on that.
Yeah, great question. So let me just, I'm going to pull up actually the part of my slides where I have it's little, what I think is an adorable cartoon, but you know, I'm not an artist, so it's really not that adorable. But here is the difference between respiration and fermentation, but happens to have both forms of fermentation on it. So here's our good friend respiration, right? We go through glycolysis, which is basically we break 6 carbon glucose down to 2 3-carbon pyruvates. In the process, we harvest out electrons. This is the decarboxylation of pyruvate, where we take electrons out again and throw one of the CO2s away. And now we've got acetyl, goes through the Krebs cycle, goes to the electron transport chain, we make ATP. Now in respiration, what happens is you go through glycolysis for sure. And then you either go to another set of reactions over here, or you go one step further and then go to another set of reactions over here, but you never go through the Krebs or the electron transport chain. So there's no opportunity to use the electrons on these NADHs for any good. You just have to dump them onto some convenient thing. And so the simplest thing is the lactic acid fermentation. I say simple because it's the first thing that you get to, and also it's what happens in our bodies. So we sometimes have a little bit more like memory of having learned it at some other point. But basically, in lactic acid fermentation, you do go through glycolysis, you produce pyruvate, and you also do collect electrons in that process. And then you just take those electrons, because they're waste, you're not going to use them for anything. You just dump them back onto pyruvate and re-oxidize your NAD. And that pyruvate with the electrons turns into lactic acid. Okay, so that part makes sense?
Yes, that one makes sense. I think it's the ethanol one that is confusing.
So in the ethanol one, what you do is you take the pyruvate and you allow it to go basically one more step, and you just remove a carbon and take some more electrons out. So you have extra NADHs, and then you dump those electrons back onto this acetate, basically. So you've generated carbon dioxide, and then the reduced acetate turns into ethanol.
Okay.
This requires a different enzyme, and I'm vastly oversimplifying it too. Okay. So sometimes it's not actually the same enzyme that does this, but nonetheless, it's still removing a CO2 and producing acetate. And it's sort of a combination of having this enzyme plus this enzyme gets you here instead of here. Does that make sense?
Yes. Is one more productive than the other in terms of their energy output? Okay.
No. And that's a really important thing to understand about fermentation. The only energy that you get to keep is here. And that produces how many ATPs?
2.
Exactly 2. 2 and only 2. The rest of the reactions, whether it's lactic acid, ethanol, or some other like series of 7 wackadoodle reactions that produces some wacky organic solvent, it's all just trying to get the electrons off of NADH, and you don't get any ATP out of that.
Okay. All right. Thank you.
Awesome. Okay. Give me 1 second, guys. I have to prevent my cat and dog from killing each other. Just a sec. All right, everyone is still alive. That's good news. Next person on my list is Alexis. Go ahead.
Hello. So I have a question on the study guide, and I think it's just kind of a general concept that I just kind of want explained one more time. But the question is, what is the relationship between metabolism and growth, and how the different metabolic strategies of microbes translate to differences in growth rates?
Yes, great question. Okay.
Thank you.
I say great question. It's like basically I'm saying, good question. to myself because I invented this question. If it's a sort of abstract concept question, you're absolutely right. So basically, what's the relationship between metabolism and growth? They're very intimately connected because growth can't happen unless you have energy and raw materials. And all of that is generated by a combination of the catabolic reactions of metabolism and the anabolic reactions of metabolism. So basically, growth happens as a result directly of metabolism. Now, when it comes to different metabolic strategies, we talked a little bit about this, and in order to sort of make the following statements, we have to really oversimplify the situation. We kind of have to hold a lot of things equal and then say, what if we just change this one thing? So let's imagine two organisms that have almost all of the same like components. Maybe they're like both bacteria, they're they have they're both gram whatever, their structures are very similar. Maybe their favorite temperature is the same, maybe their favorite pH is the same. The only difference is that one of them has the enzymes necessary to use oxygen in aerobic respiration, and the other one is only capable of doing fermentation. This is kind of the most extreme example that I could present you with. In that case, the one that can use oxygen, if you expose them both to oxygen, and let's imagine that that fermentative one is aerotolerant, so it has defenses against oxygen, not going to be killed by oxygen. But the fermentative one, every time it uses one molecule of sugar, how many ATPs does it get?
Is it 2?
Yes. And the aerobic respiration one, every time it uses a molecule of sugar, it gets like 38. And so that's, you can imagine that the time it takes to metabolize a molecule of sugar is sort of similar, right? But the difference is that when Bob over here, who does aerobic respiration, uses a molecule of sugar, he gets 38 ATPs, and Cynthia over here gets 2. Well, who's going to have more energy to grow faster? It's going to be Bob, right? The aerobic respiration one. So does that make sense?
Yes, it does. Thank you.
Yeah. Okay. Next we have, all right. So that's all the hands up I have. If other people have more questions, please ask away. Metabolism questions or growth questions that you came up with in the interim and realized you didn't ask earlier but want to ask now. Especially given, as Alexis nicely sort of segued into, they're so interrelated. Kiana, go ahead.
This is just real quick. Where do you recommend to study the metabolic pathways one more time? I feel like that's a little confusing to me.
What do you mean? Where do you mean? What to review?
Yeah. So what can give me more practice with metabolic pathways, or the flow of electrons more?
So I think my suggestion, as long as you feel like you kind of get the basic idea of what's happening, like if you sort of you understand, okay, glycolysis happens, the Krebs cycle happens, the electron transport chain happens, and you could sort of explain what's happening, like glucose is broken in half, and then there's 2 pyruvates, and then pyruvate is decarboxylated to acetate, and that goes into the, as long as you understand it at that level, then I would just review it by drawing the pictures and asking myself, where are the electrons right now? Where are the carbons right now?
Okay.
That's sort of what I would suggest. If it's more fundamental and you're like, I feel a little fuzzy on exactly what's happening in these pathways, then I would go back to the textbook and sort of this, the traditional diagrams of the steps and make sure that you feel really solid about glucose is broken down to pyruvate. Pyruvate is broken down to acetate. Acetate goes through the Krebs cycle. So it just depends on where you're feeling that need to reinforce at what level. But if you feel like I totally get all these metabolic pathways and I kind of know where all the things are, but this curveball you threw at me with electrons and carbons, I would just draw them out and then say to yourself, okay, if I was only going to draw where the carbons are, right? What would I draw? If I didn't care about enzymes or electrons or anything, I'm just caring about carbons, well, I'm going to draw 6 carbons. And then at the end of glycolysis, now I have 2 3-carbon molecules. And then I go to the next step, and now I have 2 2-carbon molecules, and some CO2 got thrown away. You're just following all the carbons. Does that make sense?
Yeah, that makes a lot more sense. Thank you.
Great question. Lauren.
So there's a question that says define electron carrier and provide examples from respiration and photosynthesis. And then in parentheses, it says hint, there are, and then in mine, it says 3, 2 in respiration and 1 in photosynthesis, but I'm wondering if I maybe added a number.
No, you did not. There are 3.
Okay.
This could be a dash. I'll make it into a dash. 2 in respiration, 1 in photosynthesis. There are 3 total.
Okay. Oh, I see. I see. So it's just, so I have in respiration, the electron carriers are NADH and FAH or FADH2.
Yes.
And then in photosynthesis, it's NADP.
Correct.
Okay.
Yeah. And then just the definition, of course, is that it's a molecule that easily accepts and then gives up electrons, right? Carries them around for metabolic pathways to use.
Okay. Thank you.
Okay. Other questions, guys? Metabolism or growth? We are now officially in the part of the class where we can, where you're invited, not required or restricted, but invited to talk about genetics. But if you have questions about metabolism or growth, feel free to ask those as well. But if you have burning questions about the genetics section, this is a good time to ask those questions. Kiara Clark, go ahead.
Hi, I'm confused on the part of the study guide where it says understand why and how prokaryotes can perform continuous replication. I know that they can perform continuous replication. I just don't know how they do that process.
Okay, so let me ask you a question. Do you understand why eukaryotes cannot?
Is it because no, I actually don't.
Okay, that's a great piece of information for me. So basically, I'll tell you, but then I'll show you.
Okay.
So first I'll tell you, the major difference between eukaryotes and prokaryotes is what? What's the major defining distinction between them that we learn in the beginning?
Eukaryotes have organelles and prokaryotes don't.
And specifically the main organelle being the nucleus that you can see. This is one of the ones that was first, the very first distinction was made on the basis of sort of size and the fact that there was this blob in the middle, it turns out to be the nucleus.
Yes.
So the nuclear envelope surrounds the DNA in eukaryotes. It does not surround the DNA in prokaryotes. And so the process of cell division in eukaryotes requires that you have to care about where the DNA is and surround it with a nuclear envelope. So you have to go, if you're a eukaryotic cell, when you go through division, you first have to divide your DNA into 2 piles and then surround it with a nuclear envelope. And now you have defined these 2 different piles, and then you divide your cell in half. But
Yes.
prokaryotes don't do that. They don't have a nuclear envelope to worry about. So they get about halfway done replicating their 1 circular chromosome, and then they start a new round of replication. And then what happens is that what gets inherited by the next generation is not 1 chromosome, it's like 1.5 chromosomes, a chromosome that's already halfway done replicating.
Okay.
And as soon as that shows up in the newly born cell, it starts doing the same thing. It's halfway done replicating, it's born that way. And so then to both the origins on each chromosome, it starts replicating those so that when it gives birth to its kids, they inherit that halfway replicated chromosome as well.
Okay, so they kind of have like 1 chromosome already replicated and then like a half one that's like waiting to be replicated.
But they're still attached.
They're like attached to each other.
Yeah. And so I'll show you the picture of this, which may help to make it a little bit more sensible. Let me see if I can just find the picture.
And they can do this because they don't have a nucleus, a nuclear envelope. That
Correct. Yes.
Okay. Okay.
Yeah. Okay, so here is in this picture, you can see this. So right here, this little purple diagram is a chromosome, 1 chromosome. And you might imagine, oh, that's what the cells inherit when they're born, but that's not true. They inherit this when they're born. It's this plus half of it's already replicated.
Okay. Okay, yeah, I think that's where I was getting confused. I thought they inherited, like you said, that whole.
I mean, they inherit this plus, it's not that they inherit this plus that. They inherit this. This is what they inherit.
Okay,
This part is the unreplicated part,
okay,
and this part is the already partially replicated part. So this is like, as soon as this, if this keeps going, which it will, it's going to produce 2. Right now it's 1.5.
okay, yeah, that makes sense. So they inherit 1.5 like that.
Yes.
Okay,
But as soon as they get it, they start replicating here and here. So they're going to produce 2 1.5s and give the 2 one of the 1.5s to one daughter and the other to the other daughter.
okay, that makes sense.
Amazing, because I feel like I explained that really poorly, but good job for following that.
Yeah, that makes sense. That makes sense.
Okay. Let's see. Chris has a question in the chat. Does that mean the perfect circle of DNA never really exists in nature? There's no, not exactly, Chris. So here are the circumstances where that perfect circle exists. For a minute after the two things separate, if we are no longer growing. So imagine, let's say, I'm just going to create a scenario. It's not the only scenario, but I'm just going to create one. You are born with this, but it's in a crappy environment. Like at the minute you're born, like suddenly we ran out of food. Everybody ate all the food before you showed up. And so the cell is like, uh-oh. And so it's best to use its resources to continue replication here, but doesn't start a new round because it's receiving information that the environment is really crappy and there's nothing to do about that. And so then at that point in time, the cell will just finish the division and probably the newly born cells will just look like this. Okay, great. I'm glad. Let's see. I need to stop share and then look at my. Okay. Kiana, you had a question.
Yeah, so real quick, right under that question that we just went over, it is understand the consequences of continuous replication. So what would be like included in the list of consequences?
Well, if you're able to start a round of replication before finishing the last one, and if you are on, like, at least if it's in log phase, if the cells as they're being born are inheriting not one chromosome, but like basically 1.5 chromosomes, then it's going to take half the time for them to finish that replication and divide again. So it cuts their generation time, at least theoretically, in half compared to a eukaryotic cell, if assuming that cell had all other things being equal, the same growth conditions, the same amount of DNA, et cetera.
Okay, perfect. So it just makes it quicker for the people, for the like cells that inherited the 1.5 chromosomes?
Yes.
Okay, perfect. Thank you.
Yeah. Okay, next we have Alexis. What's your question?
Okay, I have to scroll down and find it, but it's also on the study guide right before viruses started. But I guess the last few questions were kind of confusing to me, but mostly where it talks about horizontal gene transfer and below that where it says apply these concepts into a previous topic.
Yeah. Okay. So one of the things that was sort of like, I think, blew people's minds when they started to understand bacterial genetics better, is that horizontal gene transfer happens. But before we understood really how it happens, what was observed was that the rise of antibiotic resistance in many species of bacteria was happening faster than you could predict if you were using algorithms that were based on what we know about eukaryotic genetics. So, for example, when you look at how fast gene mutations arise and new genes and adaptations occur in eukaryotic genomes, it's based on how fast can you get mutated and pass that down to your offspring. And it was happening much faster in these bacteria, even than could be explained by the fact that they have like a 30-minute generation time sometimes. And so basically what horizontal gene transfer does is it short circuits the innovation process. Okay. It's sort of like, imagine a world in which, in the eukaryotic world, it's like everyone is siloed. And if you're in, let's say, whatever, some small town in China, you have to invent a table. And then maybe like, in Europe over there, they don't have tables yet, but they have to separately invent tables, and it takes everybody a long time. Whereas in the real world, you invent a table in China, you put it on the internet, the people in Europe are like, holy shit, that's a table. And they make the table based on what you just did. So it's kind of like that. It causes the evolution rate in bacteria to be very, very fast, because you don't have to wait as Staphylococcus aureus to invent everything yourself. You might just be lucky and someone's DNA is laying around and they had an antibiotic resistance gene, or they had a toxin gene, or they had some metabolic ability, and you get that. And now suddenly, bam, you have all this new ability that you didn't have to invent, and you didn't have to go through this long evolutionary process to get to it, or which might never ever have happened anyway, because it's somewhat random. So does that a little bit help to answer that question?
I think so. So you're like saying the evolution rate in bacteria is going to be faster because of this?
Yes, horizontal gene transfer increases the rate at which species in the prokaryotic world develop antibiotic resistance, or it causes toxins to suddenly show up in one species that did not evolve them themselves, and it's much faster. Like a toxin gene in one species could end up in multiple different lineages just very quickly. And similarly, metabolic strategies can be shared this way too.
Okay, thank you.
And then you also asked about the next prompt, which is integrate and apply these concepts to growth and metabolism. And really, the relevance here is that a lot of these things that are being passed around genetically affect growth and metabolism directly if it's metabolic enzymes. Like, let's suppose that E. coli lineage developed arabinose, the arabinose operon, and the ability to metabolize arabinose, and nobody was able to eat it before then. And then, suddenly a whole bunch of different species are hanging around dead E. coli's, and they suddenly get the whole arabinose operon, and now everybody can eat arabinose. And because metabolism underlies growth, it's relevant to that. So does that kind of relationship make sense?
I think so.
Okay. And did you have any other questions on this part?
Well, on the question below that, when it's talking about like the GFP, that's kind of what we talked about in lab, right? When you had that diagram that you were explaining to us, like, is it going to show color or not?
Yes.
Okay.
Yeah. So this is basically, if I gave you a different plasmid, not P glow, but blah, 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 P pink.
Yes.
And it didn't have ampicillin resistance. It had like erythromycin resistance. Would you be able to explain what's happening based on just general principles of how transformation works, and that these cells acquire new abilities, and extrapolate beyond the P glow?
Okay, so that's the one that you were telling us to like mess around and like make our own examples to get
Yes.
the concept right. Okay, perfect. Thank you.
Yeah, great question. Okay, let's see who else is up. Sherry, I think you were next.
I think Kiana was next. I think.
Oh, Kiana. Did you have a question, Kiana?
Oh, I think I just forgot to lower my hand.
Yeah.
But
Go ahead.
also, actually, while we're at me, I do have a question about transformation efficiency.
Yes, go ahead.
If we can just go over it one more time. I think it's just kind of hard. It seems really easy, but I feel like I haven't like grasped it.
Right. So you would like to just go through an example?
Yes, please.
Okay, great. We can totally do that. Sherry, can I just touch base with you and see, because I'm going to definitely go over transformation efficiency, but did you have a question you wanted to get in before then?
Yeah, so when we're like comparing the two different mechanisms of repressible and inducible operons, I get a bit confused on like, if, for example, you like on a test question, if it's not like super obviously specified, like what mechanism we're talking about, how is it, how can I know if that secondary sugar is present, if it's coming in as an inducer, as a co-repressor?
Right. Yeah.
Like I understand it perfectly if I know what operon I'm like looking at, right? But if I don't, it can be confusing.
So I would say this, so you can assume this. If I ask you a question about sugar metabolism, except for glucose, that operon is regulated the same way lactose is. And so you would, and I would ask you specifically, like if I was going to ask you a question about operons, I would be like, let's say we're talking about the arabinose operon. At the arabinose operon is the repressor bound under these conditions, right? So you can say, well, I know arabinose is going to be regulated like lactose, where the repressor is sensitive to the sugar, arabinose in this case, and glucose, and CAP is sensitive to glucose. And you know how CAP works and how the repressor works and how RNA polymerase works. And so it's all going to be exactly the same. The only difference is which sugar is affecting things. Does that make sense?
So yeah, so just to recap then, in like glucose obviously like constitutive, so it's repressible, but is every other secondary sugar usually inducible then?
Yes, you can
Yeah.
safely assume that for this class is the case.
Okay, thank you so much.
Like if I asked you about ribose or erythritol or mannitol or maltose or fructose or sucrose, it's all inducible. It's all has a shitty promoter. It needs CAP to have RNA polymerase find the promoter. Its repressor is sensitive to that specific sugar. CAP is sensitive to glucose. It's all the same.
Okay, thank you so much. Makes sense.
Yeah. Okay, so now let's talk about transformation efficiency. Let's just do a little, I'm going to use the example. I think I put one in the study guide. Let's find out if I did. Not sure if I did. Don't remember what my questions were. Yeah, let's use the example from the study guide. All right, so share my screen. All right, so in this, and I think I'm going to do what I did before. Oh dear. What did I just do? Oh, there it is. Yeah, okay. I'm going to share my whole screen because then I can open up a Word document and write on it. Okay. So in this example, we have a transformation reaction where we have 1 microliter of a 0.2 microgram per microliter DNA solution was added. And so the total DNA added to this tube is what? We have to do our volume times our concentration. So volume is 1 microliter, and this is our concentration. So it's 0.2 micrograms. Kiana, does this part make sense?
Yeah, that makes sense.
Great. Now we also need to know our total volume. So if we look at this, it says the total volume was 1 mil. So you see how in this example, I just gave you the total volume. Sometimes, and probably on a test, that's what I would do, because in lab and in some of the quizzes, you had to add up some numbers because you had to remember the procedure. But remember, I'm not going to test you on a test on the procedure. So I would just tell you the volume probably. The total volume is 1 mil. Okay, so now we know what the total volume is. We know the total DNA. And so now we have to figure out what fraction of that was actually used for each plate. So we used 200 microliters on each plate. So the fraction used was 200 over 1,000, right? The mil is 100 is 1,000 microliters. So we could say that this is a fifth, or we could say it's 0.2. But either way, if we want to know the total DNA per plate, it's going to be 0.2 times 200 over 1,000, which is 0.04 micrograms, if I'm not mistaken. Let's find out. I don't remember. I wrote this so long ago, I don't even remember my own question. Does this part so far make sense, Kiana?
Yeah, that makes more sense.
Okay, so now we have to know total transformations per plate. That part's easy because we just average the plates that matter. The plates that matter are the plus DNA plates. No DNA tube is a control, but it's not going to tell us how many transformations happen because no transformations happen here. We do the negative control to make sure everybody's alive on LB, and on LB TET, we do it to make sure that everyone's dead. So these are the only ones that matter. And the plates are shown here. Now, I know the plate labels are gone in this example, but we're looking for colonies. And so, we can talk more about how we figure out which plate is which, but basically the colonies plates are the ones we're looking at. And so the average of the successful transformations is 79 plus 86 divided by 2. And could I have more distractions in my world right now? I don't know. Seems impossible. If anyone can quickly do this math before me and put the answer in the chat, go ahead. 79 plus 86 is 45.5. Yes, I think you're right. 82.5, almost right. 82.5. Just kidding. Kiara, you're funny. I love it. Okay, so does this part make sense?
Yeah, this whole thing is making way more sense now that we're going over it again. It's just I felt like I was overcome.
Yeah, it is. It's overwhelming when you kind of are new to it. So you just take your transformations over your DNA and you get an answer. And again, if someone wants to do that math and put their answer in the chat, go ahead. I will try to get there as fast as I can. And then we're going to find out if I even have the same answer as I originally had. Who knows? It'll be fun. Okay, so there's my reaction. So it looks like I solved it the same way as last time. So is that helpful?
Yeah, thank you.
Awesome. Okay, guys, I need to take probably a 4-minute break because my cat just spilled a bunch of water on my electronics. So I have to go make sure that my computer doesn't blow up. So I will be back in 4 minutes to resume our conversation. We'll switch to viruses or stick with other things if people need. I'm just going to take a little pause. All right, so let's move on and talk about viruses. Also, anything else from the Unit 2 is fair game, but let's kind of make sure that we give some time to viruses. If you have questions about viruses, go ahead and ask away. If you have questions about other things, those are also welcome. Let's start with Christina.
I just have a quick question about the virus. I was watching your pre-lecture about the stranded RNA, and on one of the pictures you said it was times by 1,000, and I was wondering, what do you mean by 1,000 when it finishes up to go and exit out of the cytoplasm?
Oh, I suspect I'll have to double check because if it was the pre-recorded lectures, I may be misunderstanding what you asked and you should correct me.
I have a picture that I drew out. If you
Yeah, go ahead.
Okay. Let me.
It could mean that one virus is replicated into 1,000 viruses.
Oh, okay.
The most straightforward explanation of what you're describing.
Right here.
Oh yeah, yep.
1,000 cells, right?
Yeah, what it means is that one virus gets in, and when it replicates, at the end you have like 1,000 virus particles. Obviously, I don't know if it's exactly 1,000.
Oh, okay.
No.
But
so it's just sort of intended to indicate it's a lot.
Okay, sounds good. Because I thought like you meant like actually 1,000, but then I was like, that's impossible. They're like should be more,
Yeah, it could be. And it depends on the virus. For example, it's sometimes hard to know exactly how many because some viruses, HIV is one example, they bud instead of exploding. And so you don't get all the virus particles all at once. And then how many exactly are produced depends on how long the cell is sort of allowed to survive in that budding state. Okay. And it can be a long time. They're not healthy, but it can be a long time. And then sort of accumulating numbers. So it's not just always one number.
Okay. Yeah. Thank you.
Great question. Alexis, looks like you have a virus question as well.
Yes, I do. I have a couple if nobody wants to interrupt me when I ask them.
Go right
So
ahead.
the first one is going to be describe replication in the 4 major types of animal RNA viruses, like the plus SS RNA viruses.
Uh-oh, I lost my, just a second. There, what happened to my keyboard? Heavens above. I am so sorry, Alexis. Just 1 second while I locate all the pieces. I had to clean everything off my desk and I put places and things and I didn't realize I'd put my keyboard somewhere. Oh, there it is. Oh my goodness. This is getting recorded. That's so exciting for me. Okay, here we go. And so is this from the study guide?
Yeah, this is from the study guide. It's towards the bottom of the virus section. And then I was also confused on the questions above that one as well.
Okay. Please give me one more moment to remove the distractions from my desk. They're fluffy and adorable, but it's terrible here right now. Okay, just a second. I want to share my screen and I want to share this here. All right. So is this the question you were asking about?
Yes, it is.
Great. And then you had a second question. I will follow up with that. Yes. So describe replication. So what this means is those viral life cycles that we went through. And I'd be happy to go through them again if that's what you were asking about, but I will also point you to a really awesome resource that another student created where they made their own cartoons. And then I'd encourage everyone to make your own cartoons or follow along. So what this is asking is, you know, can you, I wouldn't ask this on a test. I'm not going to say, please diagram the whole life cycle. But knowing what that life cycle looks like will allow you to answer questions about it, if that makes sense. So, you know, that's kind of what this is getting at is, can you diagram out all those things, or do you know what's happening during the life cycle? Like, so all those cartoons that I drew, and then also have you seen this one? Let me see if I can find it. This is a much better cartoon of the life cycles than what I could draw because I have limited skill. But have you seen this viral life cycle document?
Yes, that's what I was kind of looking for. I couldn't really find the answer in the textbook, so I can look at that again, and I'm sure I will be able to figure that.
So
question
what specifically were you wondering about when you look at these diagrams? Are there things about this that you kind of want me to sort of explain more explicitly because it's not really obvious from the diagram?
No, I think I understand the diagrams. I just forgot where I found the information and I couldn't figure out where to answer that question. But yeah, I remember those diagrams. I think that will help me out a lot with that.
So this is one place. And then the other place, and I'll screen share this as well, is in our, it's not the genetics one. I think it is maybe this one. The little diagrams that I drew in the virus section. And I think it is here, yes. So the slides in this presentation here for chapter 13 that go through the different viral life cycles might be useful to you. They're not as nice as the ones that she made, but the context is what we talked about, you know, like what is the overall life cycle of the positive sense RNA, the negative sense RNA, and so on. And I'm happy to go through those guys with you if that would be helpful. But that's kind of where, and this presentation is the virus one, and it's found, let me screen share this. There we go. It's found in the weekly module for this week, right in here. So were you able to locate this slide deck as well?
Yeah.
Okay. So with all that said, so now we know where all the things are. Is there a particular life cycle that is most problematic that you'd like me to go through?
No, I think I'm okay now. I guess I just kind of wanted to make sure I was looking in the right areas for that information because it was kind of broad. So I think I'll be able to look at the diagrams and understand that question better.
Okay, great. Now let's go back and circle back to what the other thing you asked. And I'm so sorry. I was just a little bit distracted when you asked me, what was the second question? You had a follow-up question as well.
It was right above that. It was the be able to compare and contrast. Yeah, that one.
Yes. Okay, so both bacteriophage and DNA-based animal viruses are DNA-based. So you have to remember that bacteriophage are only DNA. But their difference, of course, is two things are the major differences. Structurally, bacteria have a cell wall to navigate through, and DNA animal viruses don't have to cross that cell wall. So how they get into the cell is different. Bacteriophage inject, leaving the capsid outside. DNA animal viruses enter in whole, either by fusion or endocytosis, depending on whether or not they have an envelope. So that's a big difference. The second major difference is that bacteriophage are, they infect prokaryotes. Of course, there's no nuclear envelope, so everything happens in the cytoplasm, right? The DNA is replicated in the cytoplasm. It's transcribed and translated in the cytoplasm. The viral particles are assembled in the cytoplasm. In animal viruses, the DNA virus gets in whole, right? And then it has to uncoat, and then the DNA has to get into the nucleus. And then in the nucleus, it's replicated and transcribed, and then it has the transcript has to leave the nucleus and it's translated in the cytoplasm. So those are the differences. Does that make sense?
Yes, that makes perfect sense.
Excellent. Awesome. Great question. Okay. Looks like Kiara Clark has a question.
Yes, I do have another question. It's on the study guide. It's like, know how bacteriophage can be propagated as plagues.
Oh, yes. So this is from the cultivation part. So basic thing about all viruses is that you can't just, unlike bacteria, where you can put bacteria into some like sterile medium with just food, like some proteins, some lipids, some salts, some sugars.
Yeah.
For viruses, you have to provide them whole living cells as their food. That's sort of one way to look at it. And for bacteriophage, you have to provide them whole living bacteria. So you have to infect a broth culture. And then what you do is you take small volumes of that broth culture that's been infected, and you spread it on the surface of Petri dishes at low density so that the cells that are infected are few and far between, and most of the cells are not yet infected. And that allows you to see these little spots of cell lysis that are the plaques. The plaques are kind of like a viral colony, if you will,
Oh, okay.
And so that was in I'm going to also show you where that is in the slide deck that we covered in class.
Okay, yeah, that would help a lot.
So in the slide deck that we covered in class, this is the chapter 13 slides again, and this is also in the background prep that you would have done, but it's in this section where we talk about cultivation on this slide here, where you can see that, well, I don't know how obvious it is to an untrained eye, but this whole thing is not sterile food. It's covered with this, it's opaque because it's covered with bacteria that have grown together into a lawn, except the rare cells that have been infected. And that cell has exploded. All of its neighbors got infected. They also exploded, and it sort of spread out into this plaque. And the plaque is what this hole is.
Does that mean that bacteria died right there?
Correct.
Plaque is meaning bacteria death, right? From the bacteriophage.
Yes, the clearing shows you where the plaque is, but the plaque specifically means there's a whole bunch of viruses right here, and they were all derived from the single original virus.
Oh, okay.
The same way that a colony is a whole mound of cells that originally derived from one bacterium.
Okay. So a plaque derives from one virus.
Yeah, that it's one virus that infected one cell, but then it made 1,000 clones of itself in that cell, and then exploded that cell, and then infected all its neighbors and spread. And that's why there's a circle there.
Okay, that makes sense.
Awesome. Okay. Did you have any other questions, Kiara? Okay.
No, I didn't. Thank you.
You're so welcome. All right, and thank you for the thank you for the supportive chat comments, guys. My cat is just completely, here's what it is. He doesn't care about class. He cares about food, and it's almost lunchtime, and he's like completely food obsessed. Like I've never met an animal other than my dog that cares more about food than this cat. So this is what he does when he's hungry, and I can't lock him out of the bedroom because then the dog will eat him. So that's just my life right now. Okay, more than you needed to know. What are your next questions, guys? Let's see who else is next. Okay, so Christina, you had a question?
Yeah, so I just had I just wanted to say an example for the serological test that was talked about in virus. So the serological test, I had noticed that when I got my Hep B titer boost blood drawn, I predicted that the clinic had used serological tests because the serological test is to find whether or not you have antibodies already in your own body that can be immune against hepatitis B.
Yes.
And I wanted to clarify if I'm right or
You
wrong.
are. Yeah, that's an example of a serological test. And pretty much, so serological tests are a broad category of tests that and the thing that they all have in common is that they take usually a blood sample, though it could be a different bodily fluid, but usually it's a blood sample, and they're testing for something, like something in it that can be detected. Often the way that they detect that something is with antibodies, or they detect the presence of antibodies with something that antibodies will bind to. So I just imagine it as like a little fishing line, and you stick whatever bait on it that you want. And, when you're doing a titer test for antibody levels to determine if a vaccine has been effective, then you attach something to your fishing line that the antibodies against the virus will bind to, which is usually a viral protein.
And usually if you do have the antibodies, then it will be reactive to it. Yeah.
Yes.
All right. Thank you so much.
You're so welcome. All right. Great insight. Any other questions about the virus section or any other part of Unit 2, guys? Go ahead and ask your questions. Kiana, go ahead.
So mine would be the last one, the last question, the viruses section. It's understand the structure and activity of prions and their relationship to animals, spongy form, and so
Encephalopathies, yeah.
yes.
Yes. Let me share that. Go ahead.
I just wanted to know what that was.
Okay. So do you remember the prions at all?
Yeah.
Okay. So basically, prions are, and this was, I'll also screen share the part of the lecture in class that we did, where you can also review that on your own to kind of reinforce it. But there's 3 types of things that are considered non-living acellular infectious agents. Viruses are one, and by far those are the most complex. But prions and viroids are the other 2, much simpler, but still infectious. And prions are actually a protein that, where is it? Oh, I think I put it at the end of the first day. Prions are a protein that we have in our bodies on our nervous tissue, like brain cells and nerve cells. And it's it's a harmless protein. It has a function. We don't really understand it very well, but it can misfold into a shape that is pathological. Misfolded versions, which are called PRPSC, for a historical reason having to do with, originally this was found in sheep, and the disease they had was scrapie because they would get itchy and scrape their skin off. But PRPC is the misfolded version of this normal protein that's found on all your nerve cells and your brain cells. And when it's misfolded in this form, it touches other normal ones and turns them into the bad kind. They glom together. You can't get rid of them. They junk up the cells and the cells die. It's a very slow process, actually, but over decades, you can end up with your nerve system being decimated and your brain basically wasting. It looks like it looks similar to the progression of Alzheimer's, which is possibly for a similar reason. Alzheimer's is caused by a different kind of protein that also seems to misfold and junk up the cells and kill them. But it's different than Alzheimer's because the etiological agent, the actual protein that's causing the problem, is the prion protein and not something else. So does that help to sort of clarify what that is?
Yeah, that helps a lot, actually. Thank you.
Awesome. You're so welcome. Okay, let's see what is next on our agenda. Can we talk about the one above it as well? Kiara Shepherd asked. Yes, absolutely. Let me go and share that one as well. Understand the structure and activity of viroids and their relation to plant wasting diseases. So viroids are not made of protein, they're made of RNA. And they're just a little piece of RNA. They're itty bitty. They're usually about 300 nucleotides. They don't code for any proteins. They're just RNA. Single-stranded RNA is interesting because it can function kind of like a protein enzyme because it has the ability to make hydrogen bonds and form a particular three-dimensional structure. It can have function. In fact, the early life or pre-life on this planet is hypothesized to have been RNA-based, not protein-based. So these little viroids are self-propagating RNAs. They get into a plant cell and they make more copies of themselves. And because they're presumably because they're taking over the plant's resources, the plant doesn't have the opportunity to grow in a normal way. And so it has a wasting kind of phenotype. Only infects plants as far as we know. And there's more than one actually type of viroid. They're a classification, whereas prion is one protein that we know about. But viroids there's several different ones. This is the one that infects potatoes. Does that help, Kiara? Okay, great. All right, what else, guys? What else is on your mind? We have 11 minutes before I have to run away and feed this cat before I don't know what he's gonna do. Just meow me to death, maybe. But Chris, go ahead.
I have a question outside of the test about the project ID
Oh, yeah.
thing. So I'm working on the lab report right now,
Mhm.
and the Dropbox for it, the dates for the early thing are summer dates still. It says
Oh,
August
shoot,
19th
yeah.
for early. Is there at least an early
There
extra credit thing?
is, there
Okay.
will be, and I haven't updated it just because I'm kind of going week by week. I'm behind the 8 ball, just to be very honest. I'm kind of just barely 1 second ahead of you guys. So
No problem.
because I've been having to pivot every quarter. Every time I turn around, there's something new I have to do. So not all the dates are updated, and I'm sorry about that, but I will make that a top priority since I know that you're working on that, to update that today so that you don't have to wonder when that is. But it is
perfect.
it should be accurate on the, let me
It
screen share
is
this.
on the grade book and everything.
But also on the calendar.
Okay.
So if we go here and we go to the calendar, I can just show it to you now, just so you kind of have the answer right away. You don't have to wait for me to take hours and hours and forget about what I just said I'd do. The early extra credit deadline is right here on the 28th of November. And then the final deadline is the 5th of December. So for everyone else who's paying attention or is wondering what we're talking about, this is the final deadline for the lab report. There's no penalty for waiting until the 5th, but if you submit it a week early, you get 5 extra credit points. And the reason for that is selfish. These lab reports take me freaking ages to grade. And if everybody hands them on the 5th of December, it's like Christmas before I finish grading them. So if I can get you guys to hand them in early, I reward you. And then I don't have to be like grading until the middle of the night every night for a week. So does that answer your question about when it's due, Chris?
Yep, that's perfect. Thank you so much.
Way too much more personal information. All right, let's see who else has got questions. Inessa, go ahead. What's on your mind?
So only lab 6 is due this week, not lab 7.
That's right. So let me just share this. And I think I had an incorrect date on the Canvas course shell, but it was correct in the schedule. And thank you for asking that. So as the course schedule shows, lab 7 is due on the 15th, lab 6 is due on the 8th. And I think I had it wrong in Canvas and figured that out and fixed it yesterday. If it's still not fixed, let me know. But Inessa, is that why you're asking? Because you saw the dates change? Yeah.
so.
And you were wondering like what's real, or is she just having a stroke? This is real. Yeah. So lab 7, I'm just giving you extra time because I mean, technically lab 7 is relevant to unit 3 more so than anything. So it's not as important to me that you get it done before the test. And also you have a test this week. So lab 6 is due tonight and lab 7 is due next week if you need a little extra time.
Okay, thank you.
You're so welcome. Thank you for asking that. Sharon, what's your question?
Now that we're talking about the lab, I just had just remembered a couple of questions I had about the lab.
Yeah.
Number 1, with lab 7, do you need a conclusion? Because you didn't specify that.
I do need a conclusion. Thank you for asking, Sharon. And I'm sorry if that was unclear. You always need a conclusion. So let me go and check my wording. And if I didn't put it in there, I apologize. It is a conclusion is necessary and it's the same as always. So let me go in and fix that on the lab 7 Dropbox right away.
And then secondly, you gave us the Petri dishes in inches. Do you want us to make measurements in inches and then just end up converting them to the millimeters?
Great question, Sharon. No, I want it in millimeters. And the reason it's in inches is not in the example I give. It's how do I explain this properly? The conversion of the thing on the screen I measured in inches, and then I just did a conversion based on that, but the inches go away. So it's always in millimeters. Like the plate is 100 millimeters in reality, like the actual physical plate, if you were to handle it, is 100 millimeters wide or 10 centimeters, which is the same thing. And so you need to give me the sizes of the zones in millimeters. And I gave you a way to think about how to do that. There were several ways to do that measurement from the screen for the digital part of this lab, which I assume is what you're asking about.
Yes.
Yeah. So let me just review that to make sure that it's clear. So hold on one second while I find that explanation, because I don't remember where I put it. I think it's in the lab 7 overview, perhaps. Nope, not there. Let's check lab 6. Was that a bird?
No, it's my son. I apologize for that.
Oh, my daughter makes lots of weird sounds too. I never know what's going to come out of her mouth. It's very entertaining. But I was just like, oh, do you have a cool like parakeet or something? Okay, let me see if I can find this. Lab 6.
That is if a parrot is a human, yes.
Oh, that is too funny. Okay. Let's see if these are the slides I want. They are not. Boo. Okay, maybe it's in the lab 7 background. Well, if this is hard for me, I'm sure it's really tough for you guys. Sorry. Let me see if I can get into the background. Yes, there it is. Okay. Nope, that's not it. Damn it. Well, I'll just screen share since I can't find the slide deck that I'm looking for on Canvas right now. I will just screen share what I want to show you, and then we can hopefully it'll be clear. So let me go to lab 6. Okay. So is this by any chance what you're sort of talking about when you talked about inches, this statement right up here?
Yes.
Yeah, okay. So on the screen, what you can see is a picture.
Mhm.
And if I were to measure this picture in my house right now, looking at it on my screen, it would very likely be a different size than yours because your screen is a different size than mine. So we can't actually just like measure the screen and get an answer and that's the answer. You have to convert it as a ratio of what we know is a scale bar. So just like in the beginning in lab 1, when we did measurements using a scale bar to measure cell size, we're using a scale bar to measure the zones of inhibition. So really, in one sense, the simplest way to do this is like that. So if we just dispense with the inches that were somewhat confusing, and I'll come back to that in a second, and we just say, we know that this distance is 100 millimeters. Like if I measured it right now with a ruler, it's not 100 millimeters because my screen is bigger than that. But we know that in reality, if I was to hold it in my hand, this is 100 millimeters. So that's my scale bar. Does that make sense?
Yes.
And so then I just say, well, if this is whatever this is, how many of these fit across here? And, you know, when I do that alignment, 1, 2, 3, 4, 5 of them fit across. 100 divided by 5 is 20 millimeters. So this is probably the best way to do it to avoid confusion. But if you want to be more accurate and this doesn't make sense to you or it bothers you because it isn't super accurate, you can actually take like a legit for real ruler, right? And hold it up to your screen and measure your screen and measure this. And then you can cross multiply where you know that 100 is whatever that measurement was that you took. And then X is, you can solve for X. You do a cross multiplication and solve for X.
Oh, okay.
So do you know what I'm saying?
Yes, I'm now getting it because actually I tried 2 ways and I was getting different measurements. I printed out the picture and that was giving me big numbers, like really big measurements. And then I tried the screen and that was also a different measurement. So
yes, and it will be, right? Because you have to convert it with the scale bar.
Okay, now I understand that very well.
Oh, good, okay.
And then lastly, I don't know if it's okay for me to ask this question, but on the post lab, question 5, it was asking us a question, but I was wondering, is it in relation to our lab or in relation to the number of colonies in number 4?
Let me just open up a preview, so I'm not giving the answers away, but I'll open up a preview. And this is post lab 6, right? Is that right, Sharon? It's post lab 6 that you're asking about?
No, lab 7.
Post lab 7. Oh, post lab 7. Okay, sorry. Got it. Okay, hold on one second. Post lab 7. Preview. Okay, so I'm going to screen share and you can tell me where on here. So, and you said it was question 4?
It's question 5.
Question 5.
Question 5, the second part. Question number 5.
On post lab 7?
It was post lab 6, actually.
It is post lab 6.
Post lab 6. Sorry.
Okay, yeah, no worries. I was like, but there's only 4 questions. What happened? Did you come in from the alternate universe? I know you did. It's okay. All right, here we go.
We have a lot of post labs this week, so
I know it's just a hot mess. It's my fault. I'm sorry. Okay. All right, so let's go to the preview version of this and look at question 5. The amount of DNA in micrograms that added to the plus tube was?
That was okay. It's the second one. So my question is, what are we basing our calculations on? Is it because if I'm to make the measurements whether I am closer or according to the statistics?
Yeah. No, you know what, Sharon? I have to hand grade this, and it's supposed to be, I also do a similar experiment with my online class, and I forgot to change this one so that you could put in your numbers, and then I hand grade it, and instead I gave you their options. So that's my bad. I think the simplest thing at this point, given that this is due today, is I'll just censor this one. So I'll just go through and give everyone points regardless of what they answered, because that's just my fault. That was very confusing.
And
Um,
it also translates into, sorry, not to interrupt, but to question 6, because
Yes.
it's asking about question
Yes.
5.
Yes, you're right. Yeah. Yeah,
Okay.
you're absolutely right.
Okay.
Dang it. Oh, that's so dumb.
I didn't even get to question 6 because I was like, I cannot keep going because I had 2 answers. Based on my lab, it was another answer. And based
Yeah.
on answer question 4, it was another answer. So I'm
Yeah.
like, where am
Yeah.
I?
No, it's intended to be about yours, and that's my fault. I'm sorry. I'm just going to hand grade both those questions, and everyone will get all the points basically because I just screwed it up. And I will try to remember to set out an announcement about that so people are not fretting about that instead of studying for their test. Okay. So let's see. Does that answer all your questions, Sharon, or did you have a follow-up?
No, I think that's it for now.
Awesome. Thank you for asking that and for pointing that out to me. I appreciate it. All right, Christina, what's your question?
I have a quick question about the Zone of Inhibition wiki. So I know it was due last night on Sunday, but not everyone had finished it. So I was gonna use that for my post lab 7, and I can't use that if the data is not complete. So
Right. So here's how to handle that. And this is something that I think I mentioned, but I don't know if I was very good at communicating this. First of all, the deadline for the post lab is next week, not today, for post lab 7.
oh, okay.
So you have time. And second of all, if the wiki's not complete, you still have to complete it yourself, you know, so that you can answer the questions. You don't have to complete it.
For them.
Right.
Yeah.
But you have to complete it for yourself.
Okay.
Does that make sense?
Yeah, that makes sense. Thank
Yeah.
you.
Yeah. Thanks for asking that. Okay. Other questions, guys? If you have questions about the unknown ID project, that's also fine. Thanks. Thanks, guys, for all your great questions and for coming to support your classmates. If you didn't have questions, but just wanted to hear what they had to say, go ahead and sign out if you are done. And Lauren, go ahead. What's your question?
I was just wondering if after we submit the unknown ID check 3, will you be confirming if we correctly identified our species so that we can write our reports? Or
Great question, Lauren. No, what I do for the ID check 3 is I do not check if you're right. I just check your process. I check, are you recording everything the way you're supposed to? And if not, then I tell you because it will cost you points at the end. I check if you are telling me how many species are eliminated, if you have all your images, if you are writing observations, and if your records match the record that I have, so that we can kind of make sure that everything's like looking, you know, that I can catch at this level, I can catch some sort of like really big error that will cost you points later. If I notice anything else, like you're routinely eliminating species backwards, like you get a positive result and you eliminate all the positives. Sometimes people do that on accident. If I notice like something major like that, which is sort of systemic and it's going to completely send you down the wrong path, I will say something. So if I see something, I'll say something, except about the actual species you get to. So if you mostly did it right, you recorded all the things correctly, and it seems like your process in general is correct, but for whatever reason you ended up in the wrong species, I don't tell you that.
okay. And if we get to a point where we're in like best fit analysis and we end up with like, you know, a certain number and we choose the wrong species, how much points wise, if our process is correct, but that analysis is wrong, is that like a large amount of points to miss?
In that specific scenario, you would only miss 5 points.
Okay.
But, and so because the species ID is worth 5 points out of the total, so it's really the minority, it's like 10%. It's not a huge amount. However, there are other things that are related to that can cost you points. Like if the reason that you're wrong is that you interpret a test result backwards, that will be an additional point off.
Okay.
Or it, you know, so there's like little places where you can lose points that are sort of related, but if the only thing is you did everything right, except you just picked the wrong guy for some reason, you had like a momentary lapse of reason, then that's only going to cost you 5 points.
Okay.
And you could certainly make that up if you just submit it early.
Okay. All right. Thank you.
You're welcome. All right, guys. Let's see. Sherry had a question. What's on your mind?
Yeah, about the unknown lab. So out of many of not many, but at least like 4 of the tests that I've requested so far, I made sure that they were available, but they turned out to be inconclusive.
That
Is
can
that
happen.
something that like is just
No,
at random?
it does not cost you points. It doesn't mean anything is wrong.
Cool. Okay.
You just have, and actually can help you later. So if you're feeling sad about it right now, just know that all you need to do with that information is just record it in your lab notebook. You can't use it to eliminate anything.
Mhm.
And you just go and you find a new test that looks promising and you give that one a shot. If you end up at the end having run out of tests that make sense to do, where there's some pluses and minuses, like you might still have tests that don't make sense to do, don't do those ones. But if you run out of tests that make sense to do and you still have like 3 or 4 species, you can do something called best fit that some students have already done. And you can ask me about that. And in that case, the inconclusives come back and help you.
Oh, got it. Okay. Thank you so much.
You're welcome. All right, Sharon, question.
A question still on the unknown. Just what I heard from you, it just needs some clarification. Number one, you said about eliminating the positives. If my test is negative, then the positives are not part of it. Like I'm either taking the what I do not know and then the negatives. So I am eliminating the positives.
Correct.
So what do you mean by eliminating all the positives?
Oh, just this. Okay, so it sounds to me like, and correct me if I'm wrong, let's suppose that you request a test and it comes back positive. Then you would eliminate negatives. Right.
Yes.
Or if a test comes back negative, you eliminate positives.
The positives.
Right. But some people by accident get, let's say they get a negative test result and they eliminate negatives instead of positives. They just get it backwards.
Oh, okay, now I understand. And then secondly, well, in the process of elimination, in what we are recording, do you want me to write down each species that I have eliminated down in my report for that test?
So in the lab notebook, that's not necessary, but you do have to say how many. Each time you have a test result that comes back and you interpret it and you eliminate, you have to say that eliminated 11 things, or that eliminated 5 things, or that eliminated 19 things, but you don't have to tell me who it was. In the report, however, once you read the instructions for the report, you will see that your first job is to make a flow chart. And we're going to go over this in next week's lab, not this week, because there's no lab this week. But in next week's lab, we'll talk about it. Basically, this flow chart is where you go, first test run was, I don't know, let's say it was methyl red, eliminated, and list all the species that were eliminated. Next test was eliminated, and then you list by name all the species eliminated. So in your report at some point, you're going to do that. Yes.
Oh, okay. But for now, we just count the number and then that's it.
Yep.
Okay, thank you.
You're welcome. Awesome. Okay. Any other questions, guys? If no, you can go ahead and sign out. If yes, go ahead and raise your hand and ask away.